Thank you very much for the introduction. Thank you for the invitation. Dan Geschwind had the task of prying everyone away from cheese and crackers. I have to pry you away from an entire lunch. So I'm going to have an even tougher job. It's great to be here. I want to give you a brief introduction to a new emerging activity field within neuroscience called connectomics. And I want to keep my remarks brief and simple and essentially give you a couple of messages. One is the importance of structure for understanding how biological systems work, and that's my first point. And my second will be to give you an example of an interesting structural arrangement of brain circuits that we've recently come across that we think is going to be quite interesting from the point of view of how the human brain works in an integrative sense. Before I start, I want to give you this picture here. I'm reminded by my presence here of my distant background in biochemistry. I have a degree in biochemistry, and I was very attracted to protein molecules way back when. Why? Because I was fascinated by the capacity of predicting or deducing structure, the function of these molecules from understanding the structural arrangement of amino acids and peptide chains. And that capacity of understanding the functionality of a biomolecule from the structural organization of its chemical backbone has sort of carried into my more recent thinking about the brain. This is a very different structure. This is the backbone of interregional projections of the human cerebral cortex, a map we created a few years ago. And again, it's a structure, it's a network of relationships, of anatomical pathways, and I am fascinated by our ability to predict something about the functionality of the human brain based upon this network representation. Complex systems are on everyone's mind these days. As Professor Hood said on Sunday, this is the frontier, perhaps the frontier of the 21st century for the sciences in general. And many complex systems can now be represented as networks of elements and interactions between them. And they are predictive of large emergent global outcomes, such as the commuting network that you see over here. The structure of it is predictive of whether a pandemic occurs or does not occur. The citation pattern of papers across the sciences can be used to map scientific domains and fields. And of course, this is very familiar to many in the room, the interactions among proteins captured by interactomics, protein-protein interactions, are used as predictive tools to understand something about global transitions in the functionality of cells and biological systems in general. So I very much embrace the systems biology framework and see connectomics, which I'll turn to in a moment, as the extension of that to neuroscience. Common to all these approaches is that we're using network approaches to represent and understand and model and predict complex systems. Networks consist of nodes and edges, and I'll use these terms a lot in the rest of my remarks. I wanted to show you this to set the stage. One challenge that we have in the brain is that the brain is extraordinarily dynamic, as you've seen in pretty much every talk that talked about the brain so far, including the talk this morning by my colleague John Beggs. So we have actually multiple domains of networks and interactions among neural elements that we need to grapple with in our field. One is the mapping of fairly static, stable anatomical structural connections, synaptic connections, or projections between brain regions that can be represented in a network diagram. But then on top of that, we have neural activity, which gives rise to a second set of networks, which are dynamic networks of dynamic interactions among neural elements that we can measure and deduce from time series recordings, whether they are spike recordings, EEG, MEG, fMRI, what have you. So we're always interpolating our field between a structural network, a network of static relationships, of synaptic connections, and then on top of that, dynamic phenomena that we are tracking with time series recording methods. How do we get brain networks from empirical brain data in our field? This diagram here is sort of geared towards the neuroimaging community, but it can be generalized to really any model system. In fact, I am personally working on all of these up here, the C. elegans, Drosophila mouse, and also macaque monkey. But for the moment, let's stay with the human brain. We can extract structural and functional networks from the human brain by first following a series of steps to the left here. The first step would be to postulate, to break down the human brain into individual regions or somewhat coherent structural nuclei and cortical regions, which are the nodes in our network, and then we can estimate, deduce, measure the strength of anatomical relationships between them. This will yield a network, a matrix of coefficients where every entry now refers to an anatomical coupling, typically a sparse matrix, symmetric in this case, and weighted, which tells us something about which brain regions are directly connected to each other. And that is also what I then refer to and later on define more clearly as the brain connectome. On the right, a different set of activities involving the recording of time series with a variety of methods, whether it's EEG or MEG or fMRI in the human domain, or perhaps electrode recordings of the kind that John Beggs showed, spike recordings. Those time series can be processed into functional connectivity matrices, where now the coefficients in this square matrix refer to the statistical coupling between those recorded sites and the strength of those, and that's another symmetric in this case, in this case actually a full matrix of coefficients. That's what we call a structural network. This is a functional network. Functional refers really more to brain dynamics or time-dependent activity, and we can relate these to each other using graph theory and network analysis, and that is the core of the research program of connectomics. I want to say a few words about networks and how they relate, how they are interpreted in my field. I know they've been referred to a lot in previous talks, but I thought it might be useful to do this anyway because different network measures have somewhat different interpretations in different domains of science. The simplest measure, and we've heard of it before here, is the node degree, which is simply the number of connections attached to a particular node. This might be in an anatomical network the number of connections that a given neural element actually maintains with another set of elements. And it's a very simple measure. I say simple because it's local. It's easily determined if one has a network diagram. But it's very informative, actually. It tells us a lot about how influential a particular neural element might be with respect to global dynamic effects. I leave it to your imagination which of these two nodes is more influential than the other. It's actually quite easily predicted from that. A second measure often used is the clustering coefficient, which says something about the cliquishness of a neighborhood, a topological neighborhood around a neural element. How many of your neighbors are also, how many of your friends are friends of each other? The definition of a clique is that your friends all know each other and are friends of each other as well. This tells us something about the segregation, the specialization information in a local neighborhood, and it's very informative for that reason in neural networks. Motifs inform us about perhaps potential local computational circuits within the network, which are perhaps enriched or diminished within the network compared to some random hypothesis. And then there's the important concept of distance. Obviously, not every neuron in the brain talks to every other neuron in the brain. This would be completely prohibitive with respect to the amount of wiring cost that would have to be expended. Most neurons are remote to each other and talk through intermediates. Brain regions, the same thing. We generally think that communication along short paths is important because it is less corrupted by noise, more direct, faster, etc. So we pay a lot of attention to the way short paths between two nodes in the brain are configured. Here in this case, there's a minimum path length of 3 steps going through 2 intermediate nodes. This defines what we call the topological distance between these 2 nodes in the network. Generally speaking, the closer 2 nodes are, the more directly they can interact, and the more directly they can share information and integrate information. Finally, very important part of what we do in connectomics now is to look at network communities. It's also done in systems biology and proteomics and interactomics, identifying sets of elements that are highly coupled amongst each other, more densely than expected by chance, and are more weakly coupled between each other. A very important corollary concept is that of a hub node, a node that is instrumental for maintaining the integration between communities, and that is something that I will return to later in my talk. This is a very important concept now for network neuroscience. Jointly, these measures can illuminate us about the extent to which information is specialized and segregated and aggregated together into modules, the extent to which it can be shared between modules through hub regions, and it can pinpoint particular parts of the brain that are instrumental for integrative processes, such as the hubs that I'm showing you here in this little schematic diagram. When we first defined the connectome a few years ago, we thought of it as an ohm because it is an inventory, just like Professor Hood said yesterday. Ohms are generally inventories, parts lists, if you wish. The elements, the number of elements, enumerating all of them and their relations, that's the connectome at a given level of scale. And it occurred to us back then, in fact, the issue of scale is important. There are multiple scales at which the connectome can be defined. All of them are meaningful and useful, I think. Individual neurons and synapses, all the way up to brain regions and projections between them. For various reasons, in the human, at the moment, only the macro scale of parcellated brain regions and projections between them at this millimeter scale is accessible to us, at least comprehensively accessible to us. And therefore, I will dwell on this macro scale a lot. But obviously, with model systems, we can drill down into the details of neuronal architecture in much, much greater detail. And that's where model systems become extremely important. One of the foci of connectomics, when we first described it to us, was also the assessment of individual differences. There is no such thing as a typical human brain. All brains are different from each other. They're as different as our faces are from each other. And so we are very interested in understanding what are the network differences across the population of individuals, and how do those network differences relate to individual differences we can measure in behavior, cognitive capacity, and how are they determined by genetic heritability patterns. And that is a central aim of the NIH Human Connectome Project, of which I'm a very small part. It's been underway now for 2.5 years. It's an extraordinary endeavor going along the lines of what we heard yesterday. Systems biology can achieve an overarching global goal, a large goal, many people working together, rather selflessly, I might add, tremendously driven by this common objective. Data sharing as it occurs, quarterly releases now of very, very high quality human imaging data from multiple modalities that is released online even before we have a chance to analyze it ourselves. So that is an example, I think, of a systems biology type of a big science project, of a big science approach within the field of neuroscience that I think has been quite successful and will continue to be quite successful in the future. Now let's talk a little bit about what we've known about, what we've gleaned about the human connectome so far. Before the NIH Connectome Project was a glint in anyone's eye, I worked together with a Swiss colleague of mine, Patrick Hagmann, who had developed an ingenious method to acquire human brain connectivity data using diffusion imaging and tractography to first break down the cortex and subcortical regions into coherent gray matter nodes, which can then be subdivided further to yield about 1,000 network elements. And then using diffusion spectrum imaging, inferring the trajectory, the spatial trajectory of white matter pathways, long-range projections between these brain regions, which when put together with the nodal partition, gives us a network of some complexity. We applied a variety, in fact, everything we could possibly get our hands on in terms of network analysis tools, and discovered a number of properties of this human brain network, including a broad, although not scale-free, degree distribution, which immediately flags the existence of highly connected brain regions, a small minority of them. The existence of short path length and high clustering, which is what we call the small world. The existence of structural modules, which are more densely connected amongst each other and are interlinked by hub regions. And also the tendency that highly connected nodes that have a high degree, tended to be connected to each other more so than we would expect by chance. This latter finding was something that we returned to recently. I'll come to in just a moment. As an aside, notice that when you look at this network diagram, perhaps you can see it, many of the strong projections that we find are actually short, are going over short distances. And this relates to the very great importance of the brain being spatially embedded. The brain does not exist in a virtual space like the internet might, or at least the hyperlink structure of the internet might, but it's actually physically embedded. It takes up physical resources to make a connection and to make a wire, which is a thin cylinder. And so the spatial embedding aspect of the brain can explain a lot about the way the connections are laid out and arranged. However, it cannot explain everything. Whenever we find a violation of this wiring minimization constraint, that piques our interest because it might be a feature of the architecture that actually has a very important functional meaning. And I'm going to come back to that in relation to the association between hub regions in just a few minutes. Before I do that, I want to briefly mention some of the studies we've done using connectome data and then setting the connectome in motion. This on the left here is this is the structural connectome up here. This is a recording, a movie of a recording, an fMRI recording of the brain at rest. A healthy volunteer lying in a scanner, eyes open, and not engaging any particular demanding cognitive task. And the point of this is that perhaps it reminds you a little bit of what John showed you earlier, the spin model and the critical regime. In fact, there is emerging work now that shows that the resting brain, the neural activity patterns we see in the resting brain have some of the hallmarks of criticality to them, which is a very exciting finding if you remember what John talked about earlier. What we can do with this, we can record time series. This is evidently a set of time series that's been projected onto the surface of the brain. That movie can be unfolded made into a matrix of coefficients, which you see over here. That's a connection matrix of a functional connectivity diagram. And it turns out that spontaneous activity like this, 5 or 10 minutes of that recorded in an MRI scanner, are extraordinarily informative about individual differences in brain organization and about differences in how these might relate to differences in behavior. There's a whole field now of resting state, so-called resting state fMRI and resting state MEG and EEG that has been extraordinarily productive. What we've done ourselves is we've tried to interpolate between the structural connectome, which is a matrix of anatomical projections, which is sparse and shown up here, to a full matrix of dynamic relationships between brain regions shown down here, extracted from, in this case, resting state fMRI. What we can do very nicely is this is very predictive of this pattern here. So this thing here set in motion, essentially seeded with neural activity that unfolds spontaneously, is very highly predictive of what we empirically observe, which is an indication that, in fact, resting brain activity is driven largely by the anatomy underneath. It turns out that inferring from time series observation where these anatomical projections are located turns out to be much, much more difficult and a somewhat hopeless endeavor, I think, especially in fMRI, given the nature of the signal. We can also do things like systems biology does so successfully, namely make a computational model out of this. This is our coupling matrix. We then seed each node with differential equations that describe basic ionic processes, conductances, voltages, and so forth. And we can then generate synthetic time series data in a computer, a systems biology model of the brain, if you wish, that we can compare, we can process in identical ways to the actual brain recording. We can then compare the two, and we get surprisingly good agreement between these very simple differential equation-based models and the empirical recordings. I won't say too much more about this because I'm now going to turn to the final part of my talk, which is about the violation of the wiring minimization constraint that I mentioned earlier that piqued our interest. It turns out that in many but not all networks, highly connected, centrally connected nodes, elements of the network that have many connections, also tend to connect more densely amongst each other than predicted by chance. And that's been called, evidently coming from social sciences, the so-called rich club phenomena. We are all familiar with what the rich club is. It's all of those rich guys who get together and network with each other so that they can keep their tax rates down and maximizing their influence over the rest of the network. And we set out to look for evidence of rich club organization in the human brain. I'll say what we found in just a moment. This is an example of a modular network which has hub nodes that are connecting the modules to each other. But there's no particular evidence here of this network of hubs being densely connected amongst each other. Here we've added a few connections. This might be a rich club because these nodes are not only they are not only rich in terms of the degree they have, the number of connections they maintain, but they're also maintaining many connections amongst each other. It turns out that what is interesting to us is that this is really a high-cost feature of how the human brain network is laid out, because as I'll show you in a moment, many of these connections are occurring over long distances, and they are not predicted by wiring minimization constraints. And we have argued, based on our network analyses, that these long-range projections linking rich club nodes to each other are essential for global integrative processes. So here's how the rich club is detected. In fact, it does not occur in protein interaction networks. Highly connected proteins with many partners to which they structurally bind, tend to stay away from each other, tend to not bind to each other. What you're seeing here is the density of the subgraph of all nodes with a degree of k and higher. And this is the density of that subgraph, and if you compare that to a random null model where you've scrambled up the connectivity, essentially maintained the degree sequence of the network but scrambled up all the interconnections, you're seeing that there's the actual subgraph of all proteins with more than 50 or 60 interconnections is actually much more weakly interconnected than predicted by this random null model. In a transportation network, as perhaps we're all familiar with, as we're going through air travel all the time, that's not the case. Hub nodes, Chicago, Phoenix, Los Angeles, they are interconnected amongst each other very, very, very densely. And that is actually what you can find when you subject that transportation network to a network analysis. We set out with Martin Mannenheuvel a couple of years ago to investigate whether or not the human brain has rich club organization, doing a fairly standard connectome analysis, deriving connectome maps from a series of cortical and subcortical nodes, doing streamline cryptography across the brain. And we found that there was this peak that is characteristic of rich club organization, 40%, almost 50% more connections found between high degree nodes in the human brain than predicted by chance alone. Many of these connections, just visually, when you plot them, these are the red lines over here, are made over long distances. I'll come back to that in just a moment. Some of the members of the Rich Club are familiar to people who know a lot about human cortical organization. Many of these regions have been flagged in many physiological studies as transmodal or multimodal parts of the cortex. Interestingly, subcortical regions participate quite a bit in this, including parts of the basal ganglia and parts of the thalamus. When we look at communication paths, essentially plotting the shortest paths across this network between any pair of nodes, a very high percentage, I think the number is close to 80%, pass through the Rich Club. The Rich Club itself is only about 10% of the network. So if you want to travel on a short path from A to B anywhere in the brain, you have to access that centralized subnetwork, which is the Rich Club. We replicated this study in a subsequent follow-up paper, and where we also tried to investigate these communication patterns in greater detail. Once we define our Rich Club nodes and our non-Rich Club nodes, we can also classify connections as linking Rich Club nodes to each other, or as feeding into Rich Club nodes, or as connecting non-Rich Club nodes amongst each other. So there are three categories of connections now. When we look at long projections that are more than 90 millimeters long, the majority of them turn out to be projections between Rich Club nodes. This is not the case for short projections at all. If we look at the length, the physical length of the trajectories of these fibers, and we multiply that by their density, we're seeing that the overall volume, this is sort of a very, very crude approximation of the volume that these fibers might take up in the brain, proportional to the length of the fiber. The volume that these fibers take up is disproportionately large compared to their density alone. So the brain, in a sense, made an investment, gave up a lot of volume to accommodate these fibers and to make them possible. And finally, if we take all of the short paths and we line them up one after the other, and we ask what is the probability at any given time that a particle traveling on a short path might find itself on a Rich Club connection, that probability is about 40%, which again is much higher than predicted by the density of these connections alone. This is very analogous to the transportation network, to the highway system. The American highway system is about 40,000 miles. That's about 1% of all surface roads in the US, which is about altogether 4 million miles. If you look at the number of miles traveled on highways, however, it's much greater than 1%. It's actually 25%. So highways, despite the fact that they take up, in terms of density, a relatively small fraction of the surface road system, attract traffic and do so to an extraordinary degree. We believe the same is true of Rich Club connections, sort of at the highway system of the human brain for global brain communication. My final point is that we are looking at, because we have a hypothesis now about the Rich Club as having an important role to play in integrative processes, we're looking at the ridge club in various disease conditions. We're seeing whether disease conditions such as schizophrenia, which has long been associated with disconnection for over 100 years now, might be associated with selective deficits to ridge club connections. Our first answer is that this is so. We have also replicated this. Here's the ridge club curve for a healthy control, group of healthy controls and patients with schizophrenia. The ridge club effect is diminished, statistically significantly diminished in the patient population, and also this has been replicated in 2 independent subject cohorts. We always do replication because these findings are sometimes prone to details of acquisitions, scanners, things like that. So we actually replicated these across independent cohorts done on 2 different scanners in 2 different university hospitals. So this is the selective disturbance of ridge club connections and their density. It is a start, perhaps. It is not an explanation for schizophrenia, not at all, but it is perhaps a start for understanding what might be some of the network basis for this very disruptive condition. Finally, I'm finishing up. I wanted to convince you that what we can do with connectomics is essentially take the systems biology paradigm and extend it to both structural networks of the brain and also functional dynamics on top of that. Network science, just as it's the case, I think, for systems biology, offers us not just a neat way of analyzing data, but it's actually a theoretical framework for neuroscience, and it can bind together observations in neuroscience that span different scales, going from individual neurons, circuits, all the way up to whole brain systems. The connectome is, I think, a legitimate ohm because it is an inventory list of parts and their relations, which is finite, which can be enumerated, which can be objectively verified. And just like the genome, perhaps it offers us in neuroscience a foundational data set, a data set that will not explain everything. Obviously, it cannot do that, but that will provide us with a structural basis for making sense of functional or dynamic observations, perhaps linking structural variations of the connectome to behavior. It also will inform the formulation of explicit computational models, which can not only explain and replicate empirical data, but also make new predictions, perhaps in the same sense that systems biology models have been able to do. Finally, there are many clinical applications emerging already of connectomics and all possible patient populations you can possibly think of. We're working with people in various places in Alzheimer's, ADHD, autism, schizophrenia, depression, impulsivity, which is a personality trait. We're having our hands full, and I think it's a growing and perhaps promising field. I thank you again for the invitation to be here, and I hope that I've been able to get across a few of the basic things that we're struggling with. We're looking to you, actually, systems biologists, to help us with the tools that you've developed to make sense of the data that we're acquiring at an ever increasing rate. Thank you very much.